everyone. I would like to thank Matt and Andrea and the University of Hawaii Extension for this opportunity to share the work that we have been doing over the past few years at the USDA ARS in Hilo, primarily focusing on the insect pest management research on macadamia. And I just really want to give a big shout out to the USDA ARS MacNat IPM lab team. The names are listed down below. For without them, this project would not have been possible. All right, let's start with an overview of the global macadamia production. In recent years, global production is primarily dominated by South Africa, China, and Australia, while the US ranks fifth in the total global production. Within the US, Hawaii is a primary producer of macadamia, and with small scale, limited commercial cultivation in California as well as Florida. In Hawaii, most of the production occurs in the Big Island on about 16,000 acres, which mainly consist of bearing and mature trees. In the past maybe 3 to 4 years, new young orchards have also been established on the island of Maui. In terms of utilized production, the average in Hawaii from 2014 to 2023 is about 43 million pounds, amounting to about 44 million US dollars on average over 10 years. Production is notably low in 2022 and 2023, being below average. We unfortunately do not have a current data yet in 2024 and 2025. Like any other cropping systems, macadamia production is susceptible to losses, many of which can be categorized as post-harvest crop loss. In particular, post-harvest losses in Hawaii are attributed to factors such as immature nuts, moldy rotten nuts, nut germination, and majority of it is caused by insect-related damage at 43% in 2014 and 37% in 2019. It should be noted that these numbers are underestimations as they do not include or they do not account for the losses that occurred in the field or the farms. And the three major insect pests that contribute to the losses are the tropical nut borer, southern green stink bug, and the cryptophlebium moths. All of these are direct nut feeders, so they directly damage the macadamia nuts. And in the past decade or so, the macadamia felted coccidiosis has been a major threat to macadamia in Hawaii, as infestations can decrease tree health, reduce yield, and can kill trees under high infestation levels. And in the past few years, the research efforts at the USDA ARS lab in Hilo focus on these three insect pests. For cryptophlebium moths, we've been characterizing the moth species, conducting trap refinement trials, tracking spatial and temporal trends, and which overall had informed the management trials that we have been doing. For the tropical nut borer, we have conducted monitoring and management trials. And we have also been tracking the macadamia felted coccidiosis populations and its corresponding natural enemies and initiated efforts to identify its sex pheromone that can be used for its monitoring. First, let's talk about cryptophlebium moths. There are two species present in Hawaii that attack macadamia. On your left here is the macadamia nut borer, which is Cryptophlebia umbrodelta. It is an invasive species. And on your right is the koa seedworm, Cryptophobia illipida, which is actually a native species in Hawaii. Both species have similar life cycle and damage, and although the adults can be easily identified, the larval stage of these moths are hard to identify between the two species. To better understand the pest status of these moths in macadamia, let's start with the phenology of macadamia itself. Being a tropical tree, each of these stages can be overlapping. Keep that in mind. Flowering occurs earlier in the year through April, followed by nut set from February through June, and then the pea-berry stage. And by April through November, nuts are developing to maturity, and harvesting occurs from July to December. The pea-berry stage and the immature nut stages are the most susceptible stages for cryptophlebia attacks, where the female moths lay eggs on the surface of the nuts, and then the larva bores into the fruit. And the early stage attacks of these moths can cause nut drop to occur. And if the nuts remain on the trees, the larva can bore into the developing kernel, as you can see here on the photos below. The average post-harvest damage loss estimate from this pest alone amounts to about $2.6 million annually, which is about 6% of the farm value of McNuts in Hawaii. Suffice to say, it is economically damaging. Despite these devastating losses, I have not met a lot of Hawaii macadamia growers that manage for this pest, either through chemical control or through any other means. And why do you think that is? Well, it could be for various reasons. One is that macadamia production in Hawaii is naturally low input. There's really not a lot of active pest management being done. One factor could be also labor is expensive, and especially on the state of Hawaii. And another thing that we tried to address for the past few years was to update the information on product effectiveness or getting information and what could actually work as a management tool for this pest. And upon talking to growers and stakeholders, the best approach, especially taking into consideration these different factors, was to use passive and behaviorally based management tactics for this pest. And thankfully for Cryptopluvia moths, we know that these moths rely on sex pheromones. And tortricid moths, in particular, where these species belong, can be monitored through the use of sex pheromones and can be managed also through mating disruption. And however, though, there have not been a lot of recent studies on cryptoplevia moths when I started in this position. So we started with a question: are there available commercial lures that we can use to monitor for these moths? So for this series of studies, I work with industry partners in comparing the lures they have available for tortricid moths. AlphaSense had a macadamia nut borer lure, while Tresse had their oriental fruit moth lures. All these studies were done in commercial orchards with historically high numbers of moth infestations. Our results indicate that the two moth species were relatively attracted to different lures. The invasive Macnut borer, was captured more in the Trace MNB lures, while the native Koa seedworm was more attracted to the OFM lures. We also compared two different trap colors, orange and white, and we saw that trap color did not really affect the capture of Cryptophila moths, but the orange trap captured more non-target moths than the white one. So we have been using the white trap for the subsequent studies. And the next question for us was what is the relative abundance of the two Cryptophilus species in macadamia orchards in Hawaii? So for these studies, we used trapping data and molecular analysis of larvae from infested nuts. We established that the majority of the moths are the invasive macadamia nut borer, not the Koa seedworm, which is different from the distribution in the 1980s, where the Koa seedworm was the most abundant. The native one was the most abundant decades ago. And these findings over 2 years of study are really important. Why? Because this helps us develop a more targeted strategy for the most abundant species in the system. And logically, our next question was, can these patterns be observed across the macadamia growing areas in the Big Island? So we expanded our study across several orchards in the Big Island, including Kapa'au, Pa'awilo, Kiau, Pahala, Ocean View, and Kona over 2 growing seasons. We found that significantly more macadamia nut borers, the invasive one, were captured in pheromone traps compared to the Koa seedworm across all the trapping sites. This confirmed our initial findings that indeed the most dominant species in macadamia orchards in Hawaii is the invasive species macadamia nut borer. We also found that the trends of MNB activity, or the macadamia nut borer activity, varied throughout the season across the different sites, with clear peaks in Pahala and Kona occurring in July. Activity in Ocean View was highest in March. Captures in Kapaa was highest in May, and moth activity in Kiao peaked in January, while captures remained low in Paawilo throughout the year. Moving on to the management side, now that we know that the most abundant species and we have an effective lure for it, we then can ask the question, can we use mating disruption as a management strategy for macadamia nut borer? So, what is mating disruption? As the name suggests, the main goal of this strategy is to disrupt the mating of moths by developing sex pheromone dispensers or deploying sex pheromone dispensers throughout the orchard, essentially overwhelming the area with pheromone trails that the male moths will be confused and have a hard time finding females and therefore preventing successful mating, which will then result in reduction in egg laying and reduce infestation of nuts. So for our study, we used the sidetrack MNB meso mating disruption dispensers, which were specifically developed by our industry partner for this research. We use a 32 dispenser per acre rate as a starting point in which dispensers were hung on the upper tree canopy throughout the orchard. And each treatment block was a 5 acre area as shown here. We also have a control block. The dispensers were deployed in late April and was never replenished throughout the entire study, as it is a slow release formulation, which means it will only potentially require one application per year. To monitor the moth activity, traps were deployed in the middle of the orchard blocks and checked bi-weekly from May through November. This trap served as a proxy for calling females, which means if we do not capture any moths in the traps, then we have successfully confused the males in finding the calling females. Most importantly, we also assess the egg laying rate and nut damage by harvesting nuts from the ground monthly from July to November, which is the harvesting time. We also assess nuts. Well, for this study, we only assess nuts from the interior areas of the orchard to avoid the edge effects or border effects. Now, for our results, we were able to find that in terms of average trap captures, significantly less moths were captured in mating disruption blocks. In fact, we did not capture a single macnut borer moth in the mating disruption blocks, showing a complete trap shutdown. We also captured coacidrum, though, but a lower number. For egg laying, lower rates were observed under mating disruption compared to the control. In fact, it is 50% less. But the question is, why is it not zero like the trapping results previously shown? It is possible that even though we successfully disrupted the mating of moths in the treated area, there could still be mated females from the outside area which were not treated that enter the mating disruption block. And it could also be that the portion of these eggs is coacidrum. But suffice to say that 50% reduction in egg laying is significant under the mating disruption program. Now, for the most important result for growers, right? We rated for the damage, of course. We rated for has damage, shell damage, and kernel damage, consistently showing that across all these parameters, there is less damage under mating disruption. In fact, the kernel damage was decreased about 60% under the mating disruption program compared to the control. These results are indeed promising. But we also still had to ask the question, can we confirm the results for another year? And do the parameters really differ between the orchard border and interior areas to further examine the edge effects that is commonly reported in mating disruption programs? We conducted this study at the same orchard site the following year, following the same protocol where we monitored the mouth captures as well as the injury parameters, but we categorized those parameters based on location in the orchard, interior versus borders. Based on the trapping result, this was from March to June of 2025, the average macadamia nut borer adults captured in pheromone traps under the mating disruption program were significantly lower than in the control block, both in the exterior and interior areas of the orchard. Overall, though, the moth captures are higher in the border of the orchards than in the interior. As for the egg laying or oviposition rate, we find that on average, Moth egg laying in the border rows did not differ significantly between control and mating disruption blocks during both May and August sampling dates, while egg laying in the interior portion of the orchard showed lower rates under mating disruption program versus the control in both sampling dates. Now, does that relate to damage? For the early season assessment in May 2025, the same trends were observed with damages in husk, shell, and kernel significantly higher in the significantly lower in the mating disruption than the control, but only in the interior or areas of the orchard. And damage did not differ significantly between control and mating disruption along the orchard borders. However, later in the season, in August, damage did not differ between control and mating disruption in the exterior of the orchards, while in the interior areas, only the husk damage showed differences between the two treatments. So why did we not see a difference primarily in the kernel damage and also the shell damage under mating disruption and control, particularly in the interior of the orchards, which was actually what we've seen the previous year? It could be attributed to the overall low infestation of the orchard in this year. Recorded here is less than 5% husk damage in the control, which is actually over 10% the previous year. And there's also less than 1% kernel damage in the control in this 2025 trial compared to the 2024 trial. And it is also important to note that since we conducted this trial from the same site the previous year, there's a potential reduction in the pest population that could be related to that as well. The other question is, why are we not seeing any differences at all in the egg laying and damage between mating disruption and control treatments along the borders? This is actually well documented edge effects in mating disruption programs, where the border areas are considered high risk zones compared to the interior areas, primarily due to again, mated females potentially dispersing from the untreated areas into the treated areas. And there's also a lower effective pheromone concentration or diffuse concentration at the edges of the orchards. And how can this actually be addressed? In other moth mating disruption programs, uh, the growers have increased density along the border. They have also optimized block size and contiguity of the treated area. And in some instances, using complementary controls along the edge have also been employed. Now, to summarize our results for cryptophilia research, we have now been able to develop an effective pheromone-based monitoring. We have been able to establish the relative abundance of the two main cryptophilia species attacking macadamia, with invasive species the most abundant in macadamia orchards in Hawaii. And we have shown some promising results from the mating disruption study, showing lower capture rates, lower egg laying, and lesser net damage. But we also have to really take into account the edge effect that is actually quite important to be considered in mating disruption programs. It should also be noted that through our collaboration with our industry partners, the mating disruption product is now registered in Hawaii. And now moving on to the tropical nut borer. This pest can cause significant economic losses, averaging to about 1.3 million annually, which is roughly around 3% of the farm gate value of macadamia in Hawaii. And what are these? These are bark beetles first detected in Hawaii in the late 1980s. This is a close relative to the more famous cousin, the coffee berry borer or CBB. And similar to CBB, it attacks the economically important part of the plant, which for macadamia are the nuts. Its feeding damage consists of small holes through the husk, shell, and kernel. It can take 2 to 3 weeks for kernel damage to occur after the initial attack. So there's a bit of a delayed really impact on the macadamia nuts. And field infestations are associated with mature harvestable nuts on the ground, while stick tites, the mature nuts that remain on the trees, serve as TNB reservoir. For years, monitoring is limited to manual checking of infested nuts, which is untimely for management because of that delay. And while management recommendations include frequent harvesting, which may not be practical for bigger operations, especially given the labor intensive requirements. So there really has been a need to find more practical and sustainable options for TNB monitoring and management in Hawaii. So to find a trap-based monitoring tool for TNB similar to CBB, we conducted a series of studies comparing different alcohol-based lures and different colored traps. We found that methanol lures are most attractive. The higher the methanol content, the stronger the attraction. We also did find that methanol containing denatured alcohol that can be bought from hardware stores was effective in capturing beetles, which could be a practical option for growers if they wanted to monitor for TNB. As for the color comparison, we tested yellow, white, green, brown, red, and transparent colors, finding that the transparent trap captured more beetles than any of the colored traps. And we also did a study deploying traps on the ground and the lower tree canopy. We found that the traps that were deployed in the lower canopy of trees captured more beetles than the traps that were deployed on the ground. In short, to monitor for TNB beetles, you can use transparent dial traps with window openings, as you can see here, and baited with methanol containing lures and soapy water solution for beetle retention. And we have been using this trap and lure combination to study the flight activity of TNB throughout several macadamia orchards in the Big Island for 2 years now. And we are still going through thousands of beetle samples, proving how effective the traps are. And for management tools, we also investigated or assessed repellents that can be used for TNB. We conducted studies testing known bark beetle repellents, including verbenone, methyl salicylate, and combination of both in liquid and splat formulations. For those of you who are not familiar, SPLAT stands for Specialized Pheromone and Lure Application Technology. It is a flowable wax-based matrix used to deliver, it's actually a medium for delivery of semiochemicals, including pheromones, repellents, and attractants for a steady, slow release. And it can be easily applied as dollops on solid surfaces, directly applied on plants or tree trunks. And so, yeah, we compare this type of formulation with liquid formulation of the repellents that were placed in resealable plastic bags. And this study was done by deploying repellents with methanol lures side by side in a bottle trap for a direct attraction interruption experiment. We found that all repellents significantly repelled TNB compared to methanol, with lowest captures of TNB in products with combination of both the verbenone and methyl salicylate. In our follow-up field scale repellent trial, where we actually deploy these repellents on the trees directly, our preliminary data suggests that beetles captured in bottle traps deployed in plots treated with the splat formulated beetle guard, which is a combination of methyl salicylate and verbenone, repellents were significantly less than the control. It's quite interesting why we see a higher capture in the blocks where we had methyl salicylate and verbenone pouches compared to the control. We are still conducting this study, and we will be sharing, hopefully, at least a published article um, sometime, hopefully next year. So there's several series of studies that we have been conducting to address this particular hypothesis. And these results are promising. Why? So especially, we can potentially integrate them into a push-pull strategy. Will repellents can push pests away from the crop, while the attractants can pull them away from the crops, disrupting a host finding behavior and reducing infestation pressure. And for other management trials, we tested Bovaria bassiana and found that it can cause 75% mortality under lab conditions. And I believe there are follow-up trials of Bavaria bassiana and even entomopathogenic nematodes hopefully if funding support will be provided in the future. We also tested insecticidal nets and found that exposure to these nets for more than an hour can cause more than 65% mortality, which can have a potential for an attract and kill strategy, which we actually further confirm from our preliminary trials comparing non-insecticidal net versus insecticidal net in the field, with significantly more dead beetles retained in traps with insecticidal nets compared to the control. And if you think about it, we have our attractant, we have our repellent, and we also have a passive way to actually supposedly kill the beetles. And combining all of this, we are currently exploring a push, pull, and kill strategy for TNB. As for other TNB studies, we also evaluated the impacts of predatory beetle augmentation for TNB populations using predatory beetle breeding stations, and have also conducted preliminary studies on predatory beetle releases using a drone above macadamia tree canopies. To summarize our research on TNB, we have been able to advance both monitoring and management strategies for these pests in macadamia systems. We have demonstrated that TNB population can be effectively monitored using transparent methanol baited traps placed in the lower canopy. And in parallel, we have evaluated multiple management approaches, including the use of Bavaria bassiana and long-lasting insecticidal nets, which show promise as practical control tools. Additionally, we have identified repellent compounds with strong potential to be integrated into a push-pull strategy for sustainable TNB management. And we were able to provide some technical assistance as well in getting the repellent product splat verb to be registered in Hawaii. Now, moving on to the macadamia felted coccyid, which has been a hot topic in recent years. This pest is originally from Australia. It is an invasive pest in Hawaii and South Africa. In Hawaii, it was first detected in 2005 and only found on the Big Island. It can infest all the above ground parts of the macadamia tree. It can cause branch dieback and can kill trees under high infestation levels. And for its management, horticultural oils and insect growth regulators have been known to actually work for this pest. And currently, a parasitoid is being evaluated for field release in Hawaii. Its life cycle includes egg, crawler stage, which is mobile, pupal stage, and adults with winged females and flightless females. Why is this important? Well, knowing the stage-specific behaviors is relevant in monitoring for this pest. To monitor the crawlers or flightless immatures, a double-sided sticky tape wrapped around a branch is a common approach. For adults, primarily flightless females, direct counting on demarcated sampling area on the tree branch or trunk can be done, while sticky cards deployed on the tree branches can be used for monitoring winged male adults, with yellow color attracting the most. And for the ongoing studies, especially for really trying to find a more specific trap to use for MFC, Dr. Pascal Atalor-Abidjon, who is now currently with UH Manoa, in collaboration with our colleagues at USDA, Dr. Aijan Zhang and Dr. Dong Cha, they have done some excellent work to identify the MFC sex pheromone. And in fact, they are currently conducting follow-up studies in the field. And also, this is actually in collaboration with Dr. Mark Wright at UH Manoa. And lastly, we have developed a mobile app for Android and iPhone users that provides helpful pest management information on diseases and insects important to Hawaii, including coffee, tropical fruit, ornamentals, and macadamia. And this app is actually free to download. And this is in collaboration with many colleagues from the University of Hawaii, USDA, Clemson University, and the Southern IPM Center. And before concluding my talk, I would like to sincerely thank the Macadamia Nut IPM Lab for their tremendous hard work and dedication. I am truly fortunate to work with such an exceptional and highly productive team. They have become my ohana in Hawaii, and these projects would not have been possible without them. I would also like to extend my heartfelt mahalo to our many collaborators, including colleagues from USDA, university, the industry stakeholders, and grower cooperators and grower um, associations, and of course, our funding agencies whose support and contributions have been essential in making these studies possible. And I also would like to deeply express my utmost gratitude for the opportunity to serve the macadamia grower community in Hawaii. Our partnership, trust, and continued support have made this work meaningful and impactful. And I just recently transitioned into a new role, and I carry with me the meaningful relationships and experiences built through my role with USDA. And I will remain deeply appreciative of the opportunity to have contributed to this community. Thank you all for your time today, and I would be happy to answer any questions that you may have. Mahalo.